All right, well, I'd first like to thank the organizers for the opportunity to present some of our recent work related to how the composition of the gastrointestinal microbiome can contribute to progressive SIV infection of rhesus macaques. I'm a federal employee. I think everyone knows that. As such, I never have potential conflicts of interest to discuss. Just as a little bit of background, I think everyone in this room is very well aware of the data that have shown that cross-sectional analysis of the composition of the gastrointestinal microbiome between HIV-infected and HIV-uninfected individuals routinely results in the demonstration that the HIV-positive microbiome is altered compared to population controls. But when we've looked longitudinally in SIV-infected Asian macaques, we really don't see evidence that the composition of the microbiome is becoming dysbiotic as a result of SIV infection. And so kind of compared to what people have done in cross-sectional studies of HIV-infected patients compared to HIV-uninfected controls, these are the same animals in the same cage, getting the same base diet. And what we're looking at here is the composition of the microbiome in 11 animals pre-SIV infection, and then during the acute phase of SIV infection when inflammation is really going rampant. In the green, we have the Bacteroidetes. In the pink color, we have the firmicutes. And in the blue are the proteobacteria, which include a lot of the pathobiotic bacteria that we think of as being contributing negatively to the composition of the gastrointestinal microbiome. There's a large portion of the Bacteroidetes that are Prevotella, which is seen kind of to be expanded in HIV-infected individuals. But overall, if you average the microbiomes of the animals pre-SIV infection and during day 21, you really see no evidence that there is any alteration to the composition of the gastrointestinal microbiome as a result of SIV infection. And this is based upon analysis of 16S data. You start to see subtle differences if you do shotgun sequencing, and maybe Roger Paredes is going to talk about that. And in fact, Roger has shown that if you kind of control for risk factors that led to people becoming HIV infected, it's still doing cross-sectional analysis, but it seems like risk factors that lead to people becoming HIV infected themselves are associated with alterations to the composition of the gastrointestinal microbiome. So it really becomes a question of whether or not HIV itself is changing the composition of the microbiome. But analysis of the microbiome by sequencing is really just telling us who is present and who is not present. The 16S analysis doesn't really tell us anything about the functionality of the gastrointestinal microbiome. And so one of the things that we've been doing is trying to isolate and identify bacteria that are actually translocating from the lumen of the gastrointestinal tract into peripheral tissues. And what we're looking at here is the identity of microbes that we're able to actually viably isolate from liver, from mesenteric lymph node, and from spleen of chronically SIV infected rhesus macaques. If we do the same type of analysis in SIV uninfected macaques, we rarely find any bacteria that are viable. And the red bacteria are predominantly firmicutes. We've actually tried to grow bacteria anaerobically, and in a couple of cases have been able to find a few Bacteroidetes. And the blue bacteria belong to the proteobacteria phylum. And so what we're seeing is that there's a lot of firmicutes and a lot more proteobacteria than are present in the gastrointestinal microbiome. And so there does seem to be kind of a preference for proteobacteria translocating. In collaboration with Adam Bergener, what we've been able to do is to look at the proteomics of the individual bacteria that have translocated and compared the proteome to bacteria that are still from the lumen of the gastrointestinal tract of rhesus macaques, but are not translocating. And you can see here that there's a lot of proteins that are being differentially expressed in the bugs that translocate versus the bugs that don't translocate. And if we do some sort of pathway analysis, what we find is that there's really one specific protein that is expressed by 5 of the 8 translocating bacteria that we studied, and 2 of them being proteobacteria and 3 of them, Lactobacillus, Weissella, and Kurthia, being firmicutes. So even though these are very diverse bacteria belonging to different phyla, 5 of 8 of them are expressing this cytosine-specific methyltransferase. And so what we're trying to understand now is whether or not expression of this methyltransferase is actually influencing the ability of bacteria to translocate to different anatomical sites in chronically SIV-infected macaques. But one of the questions that we really wanted to try and address is now we don't think that significant dysbiosis is happening as a result of SIV infection of Asian macaques. But the question is, well, what if we induce dysbiosis? Does that influence any aspect of progressive infection, or does it change the susceptibility of an Asian macaque to acquire SIV? Our hypothesis is really that in the context of increased microbial translocation, which we know is already caused by progressive HIV and SIV infection, if we induce on purpose and experimentally dysbiosis, we could actually further increase microbial translocation, which we think would then lead to enhanced systemic inflammation, which would enhance loss of GI tract resident and peripheral CD4 positive T cells, and of course, accelerate SIV disease. So these data were published a couple of years ago, so I'm just going to show a couple of slides to highlight what our findings were. We used vancomycin to induce a significant dysbiosis in SIV uninfected Asian macaques. And what we're looking at here are several different animals that are color-coded, and the circles are the squares are pre-vancomycin and the circles are post-vancomycin, and you can see that the first principal component here is really separating the animals based upon vancomycin treatment. The second principal component is separating the animals based upon identity. So the purple animal is up here, the green animal is here, the orange animal is here, and so on. So vanc is really inducing a significant amount of dysbiosis. And that dysbiosis looks kind of similar to what people have observed in HIV-infected individuals, with kind of a collapse of the firmicutes. And in this chronoplot, we're looking at the residual firmicutes that exist. Here are the control animals, here are the vanc animals. You see a significant reduction in the amount of Clostridiales in the vanc-treated animals, and you see a significant expansion of proteobacteria. And of the proteobacteria in controls, you see a lot of proteobacteria that are kind of diverse. You see alpha, beta, epsilon, gamma. But in the vanc-treated animals, you see predominantly a huge expansion of the gamma proteobacteria, which include a lot of the things that we talked about earlier that we think of as being pathobiotic. So the question was, if we induce this significant dysbiosis in SIV uninfected macaques and then infect them, do we accelerate SIV disease? And one of the things that we first looked at was kind of the functionality of the gastrointestinal microbiome. We looked at short-chain fatty acids. We looked at valeric acid, which is a 5-carbon short-chain fatty acid, and hexanonic, which is a 6. Both of them were significantly reduced because you get rid of a lot of the firmicutes, and in particular the Clostridiales. The loss of the short-chain fatty acids resulted in damage to the structural barrier of the gastrointestinal tract. So this is a chronically SIV-infected macaque, a colon section, an immunohistochemical analysis of tight junction protein claudin is in brown. This may be a focal area of damage to the structural barrier of the gastrointestinal tract, might be an artifact, but overall the epithelial barrier is pretty intact. And you contrast that to what happened in the animals who got treated with vanc, you see significant destruction to the structural barrier of the GI tract, a lot of infiltration of leukocytes, which would be consistent with inflammation. And when we do quantitative image analysis, you see that there's significantly more destruction to the gastrointestinal tract in the vanc-treated animals compared to the controls. But surprising to us, the animals who were treated with vanc progressed to AIDS at the same rate as the animals who were SIV infected but not treated with vanc. So even though there was a significant destruction to the gastrointestinal tract epithelium, this did not accelerate SIV disease. And so one of the things that we were then wondering is, well, how much inflammation does dysbiosis by itself cause? So again, this is looking at the composition of the gastrointestinal microbiome before vanc and after vanc, and you can see significant induction of dysbiosis in the vanc-treated animals. And now what we're looking at is how much inflammation does that vanc by itself induce? If we're talking SIV uninfected animals, pre-treatment with VANC and then post-treatment with VANC, and we're looking at the frequency of activated CD4 positive T cells that express Ki67. And you can see in all 4 animals, you see a slight but statistically significant increase in the frequency of activated CD4 positive T cells that are proliferating. So knowing that there is significant destruction to the epithelial barrier and activation of CD4 positive T cells, we thought maybe here for the animals to acquire SIV. We also went on to kind of look more globally at how much inflammation was induced by vancomycin treatment in non-human primates that were SIV uninfected. And what we did is we did gastrointestinal biopsies, colon biopsies, and then we did transcriptional profiling by nanostring, which is a targeted reverse transcriptase PCR of about 800 genes of immunological interest. So it's kind of a cheap way of doing a transcriptional analysis. So what we're looking at here is a PCA plot, dimension 1 versus dimension 2. The purple circles are pre-VANC, the blue circles are post-VANC, and you can see that globally there wasn't really a significant alteration to the immunological transcription profile of the colonic cells before or after VANC. But we did kind of look at genes that were differentially regulated. There were about 29. And if you do pathway analysis on that 29 genes, the first pathway that lights up are toll-like receptor signaling. So we do think that just by itself, vancomycin is inducing low level of inflammation. The next question we wanted to ask is whether or not the dysbiosis that we induced, does that make it easier to acquire SIV? And so we started doing low-dose interrectal challenges in animals that were treated with VANC versus controls. And we used Brandon Keel's barcoded SIV MAC239 so that we could look at the number of transmitter founder viruses. So this is a virus called SIV MAC239X. What Brandon does is introduce barcodes into the polymerase gene, and they're synonymous mutations. So it doesn't change the functionality of the virus. It just allows us to be able to identify the number of transmitter founder viruses. And Brandon has shown that each one of these barcoded viruses replicates efficiently and kind of equivalently both in vitro and in vivo. And so what we started doing was low-dose interrectal challenges in animals that got vancomycin. We titered the virus such that about every time 20% of the animals would get infected. And you can see after 10, it's actually after 11 challenges, all of the control animals got infected. We then treated animals with vancomycin for 1 week, then challenged them with SIV MAC239X and looked at the number of challenges that it would take to infect all of them. And we went down to about 6. So after 6 challenges relative to 11, we infected all of the animals with SIV MAC239X after being treated with VANC. So this didn't really achieve statistical significance. It was close. But what we were able to then look at is how many transmitter founder viruses there were after the animals actually got infected. So you can see here are the controls, the number of transmitter founder viruses on the y-axis, and all but one animal that was a control got infected with 1 and only 1 transmitter founder virus. 1 animal got infected with 2. Now, if we contrast that to the animals who got vancomycin, you can see that 4 of them got infected with 1 and only 1 virus, but 1 of them got infected with all 10, another got infected with 6, 5, and 2. And so that ended up being significant, statistically significant, that there were more transmitter founder viruses in the vancomycin-treated animals compared to the controls. So just to kind of wrap up, VANC-induced dysbiosis is insufficient to accelerate SIV disease progression. VANC-induced dysbiosis is sufficient to induce low level of GI tract inflammation. This barcoded SIV MAC239 can be used to assess the number of transmitter founder viruses and the proclivity to become SIV infected intrarectally. And dysbiosis that we induced by vancomycin results in increased susceptibility to acquisition of SIV with higher numbers of transmitter founder viruses. So we know that risk factors for HIV acquisition are associated with a dysbiotic microbiome. And so we kind of think that maybe therapeutic interventions to reduce dysbiosis may decrease susceptibility to HIV acquisition via receptive anal intercourse. So I'd just like to close there and thank all of the investigators who were important for this work to be done. First and foremost, Alex Ortiz, who is a phenomenal staff scientist in my lab that really led the projects on how dysbiosis influences disease progression and influences susceptibility to SIV acquisition. Jacob Flynn is a graduate student in the lab who has been growing bacteria from different anatomical sites and categorizing and characterizing them by proteomics with the help of Adam Bergener that I mentioned earlier. All of the microbiome work that we've done is in collaboration with Yasmin Belkade with help bioinformatically from Marion Quinolins. I mentioned that I had the SIV MAC239X from Brandon Keel at NCI Frederick. I think everyone knows that the immunohistochemical analysis that I present is always done in collaboration with Jake Estes, and none of these studies would be possible without a very dedicated animal care staff at the NIH Animal Center. And I'd finally like to thank you for your time and attention and be happy to take any questions.